I'm Tomiris Nabiyeva and I'm a student at Maastricht Science program. Recently, I have completed my Bachelor thesis research on the topic of synthesizing and characterizing osmium novel compounds for anti-cancer applications. Cancer is a massively detrimental disease that has one of the highest mortality rates. It includes malfunctioning of the cells, their rapid division and multiplication that spreads in neighboring organs and eventually leads to death. There are three main treatment strategies existing today: chemotherapy, radiotherapy, and surgical removal of the tumor. Chemotherapy today includes treatment with a drug discovered by Rosenberg in 1965, cisplatin. Since then, it has become a gold standard for all the following anti-cancer agents, mainly due to its high levels of cytotoxicity, or in other words, its incredible ability to cause cell death. Due to its small size, cisplatin crosses the cell membrane easily. Inside the cell, it enters the nucleus and interacts with DNA. You can think of DNA as two dense partners. When cisplatin enters the nucleus, it becomes positively charged and attracts the negative part of DNA, sealing one of the partners and ruining the whole dance. Imagine that this is your beautiful garden which you're trying to maintain. Using cisplatin against cancerous cells is the same as using pesticides in this garden. Because it is not selective, it will equally harm the flowers that you're trying to maintain and the weeds that you're trying to get rid of, in the same way as cisplatin both kills the cancerous cells and the healthy ones. Another phenomenon that was observed is that the cells of the patient who was treated with cisplatin for a prolonged time can develop a resistance to it. Therefore, in the pursuit of finding an alternative to cisplatin, thousands of other platinum-based compounds were made and tested. Carboplatin and oxaliplatin are the two agents that had some improvement and were approved for the treatment in combination with cisplatin. However, they do not completely overcome cisplatin's issues, so the search continued among the other metals. Ruthenium was the next most promising candidate and was studied extensively. Many of the ruthenium complexes had good to high cytotoxicity levels, and some of them even went to clinical trials. One particularly interesting compound is RAPTA-C, which was not cytotoxic at all when tested in a lab, but when tested in a living organism, it damaged tumor cells selectively to the same extent as cisplatin. The discovery of RAPTA-C inspired research at MSP and directed it towards the ruthenium-based complexes. Many different combinations of compounds were synthesized and tested. But what about other metals, like osmium? Osmium is in the same period as platinum and in the same group as ruthenium. It has many attractive properties and a lot of potential. My research consisted of making 3 new compounds that could potentially have anti-cancer properties. Furthermore, they were fully characterized and tested for the stability in DMSO. Synthesis of these compounds consisted of a procedure with 3 chemical reactions. This procedure is like a recipe for a pizza. First, you need to make the base for a pizza. My base was the starting dimer synthesized in the first reaction. Then, to sort of make an intermediate pizza, you need to put the sauce on your base. In the same way, I attached the desired substituents to my starting dimer and synthesized the intermediate. And finally, you can add any toppings that you like to your pizza. Similarly, my intermediates were decorated with icogermanium or tin dichloride, which gave the final compounds. All the structures were confirmed by NMR techniques. Depending on the environment surrounding a particular atom, the peaks on the graph appear at different positions and as various shapes. Therefore, from such spectra, it is possible to identify how the atoms are organized in a molecule, or in other words, NMR techniques aid in confirming whether you have the desired structure. When I was trying to get my desired intermediate, I noticed that the reaction actually produces a mixture of compounds. From analyzing the NMR spectrum of the mixture, I was able to identify the second product to have this structure. For this reason, the reaction was performed twice in 2 different solvents, acetonitrile and dichloromethane. However, in dichloromethane, The mixture of compounds was also formed with a desired structure and a dinuclear system like this. It was decided to separate the second mixture and continue the synthesis only with the desired intermediate. However, upon the separation, the liquid crystallized at room temperature and I obtained these beautiful crystals, which subsequently could be analyzed and their exact structure was obtained. Therefore, the other mixture prepared in acetonitrile was used for the subsequent synthesis. This mixture was not separated and was reacted directly with germanium or tin dichloride. This proved that the separation step is not crucial for the reaction to work, and not doing it actually simplifies the synthesis and improves the yield. To not bore you with a lot of graphs, I will just say that all the final compounds were completely characterized using all the analytical techniques, as well as their structures were confirmed by the spectroscopic methods, and all of these graphs and spectra can be found in the thesis itself. Finally, it is important that the potential future drugs are stable in DMSO solvent. DMSO is a solvent that is used for cytotoxicity tests in the lab, and furthermore, DMSO is used for the intravenous injections of the drugs to the body. Imagine that you painted a masterpiece, and you know that it's going to be delivered by sea across the world. To make sure that it will reach its destination safely, you need to test it whether it's stable in moisture conditions. The same is with our anti-cancer agents. To be safely delivered to the cells through the blood, they need to be tested in DMSO solvent. It was observed that the most stable and unreactive compound was a trichloroethanate salt, while the least was trichlorogenol neutral compound. Such neutral analogous ruthenium structure also indicated similar results and converted into its inactive form when dissolved in DMSO. The research was successful and very informative. Many conclusions were drawn from it about the synthesis and the properties of the final compounds. All the complexes were fully characterized, and hopefully in the future tests they will exhibit high levels of cytotoxicity. Ooh! I hope you enjoyed my video. To make it more accessible, I have simplified certain elements of it. But for better understanding, you can always refer to my thesis report. And bon appétit!